When we pick up in chapter 25 in the second half, we're going to start talking about calcium ions, more specifically about the calcium and phosphate ions. So calcium ions really play a critical role in many roles of the body. As you've learned, they're one of the main components of the bone tissue, this osseous tissue, giving bone its hardness and resistance to compression. About 99% of the total calcium ions in the body are located in the bone as part of those hydroxyapatite salts. Calcium ions are also required for muscle contraction, the plateau phase of the cardiac action potential, intracellular signaling, blood clotting, neuronal synaptic transmission, and many more. In addition, they have another important role, the excitation of neurons. When calcium ion levels rise, neurons become less permeable to sodium ions, and so the reverse is also true as well. As calcium goes down, neurons become more permeable to sodium. And it's impossible to discuss sodium without also discussing phosphate, and that's because they're found together in the hydroxyapatite crystals. The inorganic portion of bone is not able to be built unless both calcium and phosphate are present. And so for this reason, they're regulated in tandem by the same hormones. Phosphate ions have other important functions like the integral part, phosphate is one of the integral structures of adenosine triphosphate, ATP. You need phosphate for adenosine triphosphate, obviously. This section, we're going to talk about the hormones and mechanisms that control phosphate and calcium. And then we're going to look at calcium ions, exploring what happens when imbalances occur. Now, calcium is going to be regulated in the ECF at about 8.7 to 10.4 milligrams per deciliter, and it rarely varies from this range by more than a few percentage points in either direction. This is a strict control thanks to the compensatory mechanisms that occur at three main sites: bone tissue, distal tubules, and collecting systems of the kidneys and small intestine. So simply put, when the level of calcium ions falls, when the level of calcium ions in the extracellular fluid falls, calcium ions are going to be released from the bone by osteoclasts, and they're going to be reabsorbed from the filtrate in the kidneys. And so more are going to be absorbed from the ingested foods and liquids in the small intestine as well. Conversely, if calcium ion levels rise in the ECF, calcium ions are deposited by osteoblasts into the bone, and then fewer ions are reabsorbed by the filtrate, and fewer are absorbed in the small intestine. However, while calcium is very strictly regulated, phosphate is not nearly as strictly regulated, as tightly regulated. The extracellular phosphate ion concentration really varies throughout the day. Generally, the body maintains a slightly lower phosphate ion concentration than calcium because these cells require a certain number of free calcium ions in their cytosol. When phosphate ions are present in large numbers, they bind to calcium and form a solid calcium phosphate salt, reducing the number of free calcium ions. So the body has to excrete more phosphate ions and retain more calcium ions. And the two main hormones involved in these regulations are parathyroid hormone and vitamin D3 in the form of calcitriol. However, there are other hormones that influence this balance, like calcitonin, cortisol, and estrogen. But if you guys remember, parathyroid hormone is going to be released from the parathyroid glands, and this is in response to a decrease in calcium ion concentration in the blood. The parathyroid hormone triggers osteoclast activity, and so also triggers calcium ion reabsorption in the kidneys. And then in addition to this, it decreases the amount of reabsorption of phosphate ions. Another important effect of parathyroid hormone is the activation of vitamin D3. And vitamin D3 is a potent stimulator of calcium ion absorption by the small intestine. D3 also assists parathyroid hormone in increasing osteoclast activity, and calcium ion reabsorption from the kidneys. With regard to phosphate ions, D3 triggers their absorption from the small intestine. And so this increases the concentration of phosphate in the ECF in contrast to the effects of parathyroid hormone, which lowers their level. This helps to ensure that the level of phosphate never falls too low in the extracellular fluid. And unlike changes in phosphate ion concentration, any small change in calcium concentration can have immediate dramatic effects on different body functions. Increases and decreases in blood calcium ion concentration, called hypercalcemia and hypocalcemia, can lead to devastating effects. And we start with hypercalcemia, which is defined as a plasma concentration of calcium ions above 10.5 milligrams per deciliter. This is typically due to hyperparathyroidism, but may also be caused by certain cancers, excess vitamin D, certain bone disorders, or renal failure. And hypercalcemia has wide-ranging effects on multiple organ systems. The most immediately noticeable are going to be on the nervous system. As you guys remember, an elevated calcium ion concentration makes neurons less permeable to sodium, which diminishes their ability to depolarize. So this decreases their activity, leading to mental sluggishness. Gosh, I think I'm mentally sluggish. Mental sluggishness, reduced reflex activity, and weak muscle activity, because these muscles are not able to be stimulated by motor neurons. And hypercalcemia can affect other tissues as well. Calcium ions are responsible for the plateau phase of cardiac action potential, which lengthens and strengthens the heart's contraction, right? But if there's too much calcium, the plateau phase is shortened, making the contraction shorter and weaker. And hypercalcemia can cause a decreased appetite and constipation because there's a decrease in activity of the gastrointestinal smooth muscle, but it can also cause kidney stones, bone pain, and frequent urination. All around a bad time. Hypocalcemia, this is going to be when the blood calcium level drops below 8.7 milligrams per deciliter, and this is generally due to a decrease in parathyroid hormone, or if parathyroid hormone isn't working, like when renal failure occurs, or if vitamin D is deficient, although there are many other factors that can contribute. And hypocalcemia has the opposite effect on neurons from hypercalcemia. These neurons become hyperexcitable as more sodium enters and depolarizes the neurons, stimulating those action potentials. When hypocalcemia is severe, there's spontaneous neural firing, leading to a repetitive stimulation of these skeletal muscles and tetanic or sustained contractions. The characteristic carpopedal spasm of hypocalcemia, you should see in the next slide here. This is that carpopedal spasm. In addition to this, the plateau phase of the cardiac muscle cell action potential is lengthened, leading to overly long and overly strong contractions. And a calcium ion concentration below 4 milligrams per deciliter is usually lethal because of these and other cardiac effects, and the tetanic spasm of the respiratory muscles. So this is not any better than hypercalcemia. Now, I said those were the big three, four if you include phosphate, but there are other ions that are crucial for human physiology, like chloride and magnesium. Chloride has a variety of functions, with a high concentration in the ECF, making them important osmotic particles. Chloride also is a critical component of some other processes, like the production of hydrochloric acid. Chloride ions are involved in the secretion of newly formed bicarbonate ions when red blood cells are made. And the concentration of chloride ions in the ECF is almost determined entirely by the kidneys, where reabsorption of these ions is largely coupled to that of sodium ions. Chloride does tend to follow sodium passively from the filtrate to the ECF due to their electrical gradient. In addition, several transporters move the two ions together. And you got magnesium ions that are critical for multiple cellular processes like activating enzymes, making up an important component of bone tissue, with about half of the total magnesium being located in the bones, with the rest of it residing in the cytosol. And for this reason, the kidneys are excreting the majority of the magnesium ions that are ingested. Maintaining a low ECF concentration of magnesium is critical because if it gets too high, it blocks the entry of calcium ions into cells, which in turn leads to hypercalcemia. Now we're going to look at a different set of ions, hydrogen ions and the various bases like bicarbonate ions. And normally we're trying to shoot for a pH in the human body between 7.35 and 7.45, very slightly basic. And it has to maintain the slightly alkaline range because even a slight deviation has disastrous consequences for our cells. And it's regulated through a number of mechanisms. The most important are the respiratory system, the urinary system, and the body's buffer systems. And there are two types of buffers, the chemical buffers and the physiological buffers. Basically, chemical buffers are chemical systems that work to buffer the fluids in the body, and physiological systems use organ systems that work to buffer these fluids. So we're going to look at these in this section here. Now, acids and bases in the body really come from two sources. Those that are formed as part of the normal metabolic process and those that are ingested as part of the diet. And we've seen how the body produces multiple metabolic acids. The biggest source of metabolic acids is carbon dioxide, the obvious product of glucose catabolism. If you guys remember, carbon dioxide itself isn't an acid, but when it reacts with water, it forms carbonic acid, H2CO3. And this then can dissociate, it'll break apart to form a hydrogen ion and a bicarbonate ion. And carbon dioxide is known here as a volatile acid because when it's in solution, it is able to change states from carbonic acid back to carbon dioxide gas. And it allows carbon dioxide to be eliminated through both the lungs and the kidneys. The body has other metabolic acids like lactic acid as a byproduct of glycolysis, uric acid produced by breaking down nucleic acids, intermediates of the citric acid cycle are acids, acidic ketone bodies are produced from fat breakdown, and these acids are not volatile acids, these are fixed acids because they cannot change from a gaseous state from a liquid state to a gaseous state, and so they all have to pass through the kidneys. And the human diet tends to be the source of both acids and bases. There's protein-rich food that contains amino acids, fatty-rich food which contains fatty acids, fruits contain acids like citric acid. Some fruits, fruits and vegetables, contain small amounts of base ions, especially bicarbonate. And although these foods do contribute the same amount of acids and bases, they represent an exceedingly small minority that have essentially no effect on overall pH, with the majority of the acids and bases coming from the body's own metabolic reactions, not from outside sources. Before we get too deep into buffer systems, let's review a few basic definitions from chemistry. A strong acid is going to be one that releases most of its hydrogen ions in water, which lowers the pH of the solution. A weak acid releases relatively few hydrogen ions and has a much smaller impact on pH. The same is true for bases. A strong base removes great numbers of hydrogen ions, increasing the solution's pH, and a weak base binds relatively few hydrogen ions. So, with all those in mind, let's talk about the chemical buffer systems. Chemical buffer systems consist of a weak acid and its conjugate weak base functioning to resist large swings in pH. So when a strong acid is added to a solution, normally the weak base of this buffer system will bind to any released hydrogen ions and remove them from this solution. The same is true in the opposite. If a strong base is added to the solution, the buffer system weak acid releases its hydrogen ions to bind those base ions. Both of these situations work to minimize pH change and maintain acid-base homeostasis. The buffering capacity that we're talking about here for body fluids is called the alkaline reserve. When the alkaline reserve is depleted or it runs out, acid-base imbalances then occur. And there are three main chemical buffer systems in the human body. This basically explains exactly what I just said with the weak acid and weak base buffer system here. If you don't understand, you can pause and read, but basically they both just work to minimize the change. You see, normally this would change it to a very acidic pH, but with a buffer, there is a minimal change. The same is true inversely with a slightly basic pH that would normally become very basic. Like I said, there are three main chemical buffer systems. There's the carbonic acid bicarbonate ion buffer system, the phosphate buffer system, and the protein buffer system. The first one we're going to talk about is the carbonic acid bicarbonate ion buffer system, and it is the most important one. This consists of the weak acid, carbonic acid, and its weak conjugate base, the bicarbonate ion. The source of one of these sources of carbonic acid is the reaction that we described earlier, where carbonic acid or carbon dioxide reacts with the water in red blood cells and it produces a carbonic acid. When the carbonic acid breaks apart, it yields one hydrogen ion and a bicarbonate ion. And these bicarbonate ions are also going to be found in salts, especially sodium bicarbonate. That's normally baking soda. Normally, sodium bicarbonate completely dissociates in solution to form sodium and bicarbonate ions. But how do carbonic acid and bicarbonate ions buffer a solution? But how is it that this works? So if we look at this, if we add an acid to a solution that is buffered by sodium bicarbonate, you see that the hydrogen ion forms from the strong acid that is going to be accepted by that bicarbonate ion, forming carbonic acid. And since carbonic acid itself is a weak acid, it does not as readily release hydrogen ions, so it minimizes the pH change. Carbonic acid similarly buffers a very basic solution. If you see, if you dump sodium hydroxide into a solution that is buffered with carbonic acid, the bicarbonate, it releases, this carbonic acid releases its bicarbonate and its hydrogen ions, and the hydrogen ions from this carbonic acid bind to the hydroxide ion that is released, forming water and a molecule of sodium bicarbonate. And the bicarbonate ion is a weak base, so it will not remove a significant number of hydrogen ions from the solution. So it's simply the presence of these weak acids and weak bases minimizes changes in pH that would normally be very drastic. Now the phosphate buffer system, this is the second one, it consists of a weak acid, dihydrogen phosphate, and its conjugate weak base, hydrogen phosphate. So as in the carbonic acid bicarbonate ion buffer cycle, the acid and the base here balance any excess hydroxide and hydrogen ions respectively, working to keep the pH stable. Now the concentration of hydrogen phosphate is too low in the extracellular fluid, with only about 8% of the bicarbonate ion concentration. So that doesn't make it an important buffer here, but it does have a higher concentration in the cytosol and in the filtrate within the kidney tubules. So it is a much more important buffer in these locations. And then you have the protein buffer system, which, because there's a huge variety of proteins, especially in the cytosol of our cells and in the plasma, these proteins can be composed of strings of amino acids, each containing a carboxylic acid group. In solution, some of these carboxylic acid groups are going to ionize their weak conjugate base form, allowing them to act as protein buffers. When hydrogen ions are added to the cytosol, they can bind to these bases and be removed from the cytosol, preventing large swings in pH. And there are many important intracellular protein buffers. About 60 to 70% of the total buffering capacity takes form in protein buffers of the cytosol. An important one is hemoglobin inside red blood cells. Hemoglobin acts as a buffer for hydrogen ions formed in the dissociation of carbonic acid. Now, those chemical buffers basically respond to changes in the hydrogen ion concentration of the fluids to maintain normal range, right? But for these systems to work, the body fluids themselves must contain adequate numbers of base ions, especially bicarbonate ions. And so the body must also have itself a way to rid excess hydrogen ions in order to maintain this normal, slightly alkaline pH. And there are two systems: the respiratory system, which controls the amount of volatile acids in the extracellular fluid, and the urinary system, which works, which both of them work to control the amount of fixed acids and bicarbonate ions in the ECF. Now, these systems also work to correct hydrogen ion imbalances, and so for these reasons, these two together are referred to as a physiological buffer system. And if we look at the respiratory effects, you see that carbonic acid in the body fluids, right, is formed when carbon dioxide hits water. The lungs directly control the amount of CO2 in the blood, so it follows that the lungs also control, to a significant extent, the levels of carbonic acid and hydrogen ions. Under normal conditions, CO2 levels are expired by the lungs to match the amount of CO2 produced by metabolic reactions. That's why your respiratory rate doesn't remain constant. When you're physically active, there's glycolytic catabolism and oxidative catabolism occurring more rapidly, and so more CO2 is created. And so your respiratory rate has to increase to match the higher level of CO2 production. The opposite is true. When you're sedentary, you generate less CO2 and your respiratory rate should decrease. Now, it also influences a number of bicarbonate ions in the plasma. And if you guys remember, the reaction between CO2 and water occurs rapidly in red blood cells due to the presence of the enzyme carbonic anhydrase. When carbonic acid is formed in these red blood cells, much of it dissociates into bicarbonate and hydrogen ions. The hydrogen ions that form bind to hemoglobin, which buffers the cytosol of the erythrocytes, the red blood cells. And these bicarbonate ions that are formed, however, are going to be transported into the plasma where they work to buffer fixed acids. The kidneys work with the lungs in two ways to determine the number of hydrogen and base ions in the blood. First, the kidneys excrete fixed acids that the lungs cannot excrete, like lactic acid, ketones, phosphoric acid, ammonium. About 25% of the body's hydrogen ions are lost in this manner. Secondly, the kidneys contribute to the acid-base homeostasis by controlling the amount of bicarbonate in the blood. Generally, the kidneys reabsorb all bicarbonate into the filtrate, or from the filtrate, but recall that there are no true transport mechanisms to move it from the filtrate to the tubular cell. So, there are more roundabout methods for reabsorption that are needed. In addition to this, the kidneys can actually manufacture new bicarbonate when the pH of the blood falls, and it will secrete these bicarbonate ions when the pH of the blood rises. We'll look at that next. Now, the proximal tubule of the cell, the proximal tubular cell, is going to secrete hydrogen ions from those fixed acids by a secondary active transport process. If you remember, this process is going to be coupled to bicarbonate ion reabsorption. So the secreted hydrogen ions bind to any bicarbonate ions in the filtrate to form carbonic acid. Carbonic anhydrase then turns this carbonic acid into carbon dioxide and water, both of which are able to enter the kidney tubule cell, and then inside the cell they react again with carbonic anhydrase, forming carbonic acid, which again dissociates into bicarbonate and hydrogen ions. The bicarbonate ion then leaves the cell, entering the blood, where the hydrogen ion is going to be recycled as it is resecreted to bind to another bicarbonate in the filtrate and just do it all over again. This process is going to reabsorb nearly 100% of the bicarbonate ions in the filtrate, and it's going to secrete a large number of hydrogen ions. There is another process that occurs there where they actually form new bicarbonate ions. We'll talk about that with the glutamine here in just a second. So the first thing that happens is glutamine enters a proximal tubular cell and undergoes reactions that yields 2 ammonia molecules and 2 bicarbonate molecules. After that, you have 2 molecules of ammonia that combine with hydrogen ions, producing 2 acidic ammonium ions. Ammonium, not ammonia. The 2 ammonium ions are then secreted back into the filtrate for excretion in urine, while the bicarbonate ions that are just been created go into the interstitial fluid and then into the blood. So you see this glutamine comes in and it breaks into ammonia, and then ammonia becomes, ammonium. But moving on, apparently it didn't want me to talk about that too long. But the major acidification of the urine is going to take place in the late distal tubule and the collecting system. And this is going to happen in cells called the intercalated cells. These cells have primary active transport pumps in the plasma membranes that work to pump hydrogen back into the filtrate. In a figure you're going to see here in just a second, I want you guys to notice that the source of the hydrogen is actually going to be carbon dioxide. So if we look at the secretion of hydrogen ions, the first thing we have to look at is this the starting thing here is this carbon dioxide. It's going to diffuse across the blood and interstitial fluid into the tubule cells following a concentration gradient. And so we get it into the tubule cell. In the distal tubule cell, CO2 and water are going to be undergo a reaction in the presence of carbonic anhydrase. So these two get combined and become carbonic acid. And this carbonic acid is just going to dissociate into hydrogen and a bicarbonate ion, right? And so this newly formed hydrogen ion is going to be transported out into the filtrate via active transport pumps, including one that pumps out hydrogen and pumps in potassium. Do note that these hydrogen ions are not going to be recycled like the ones in the proximal tubule. But what happens to this newly formed bicarbonate ion? Well, this newly formed bicarbonate ion flows out into the bloodstream. It's just transported across the tubule cell into the interstitial fluid and the blood. Normally, these buffer systems ensure that pH remains in a narrow range, even when acids or bases are added or lost. But these buffer systems can be overwhelmed by a significant loss or gain, and they'll fail, resulting in a pH imbalance. The two basic types of pH imbalances are acidosis and alkalosis, and the body attempts to correct both through a process called compensation, which involves both respiratory and or renal compensation. And compensation does bring the pH closer to the normal range, but it's not a permanent solution. Rather, the permanent solution lies in correcting the cause of the imbalance. And most of these pH imbalances are going to be secondary to other causes like respiratory disorders, kidney failure, GI problems. And so for this reason, treatment is generally aimed at the primary cause, and when that's resolved, pH returns to normal. Now, acidosis is defined as a bodily fluid pH of less than 7.35. Note that this is acidemia. This is not acidemia. Acidemia refers to low blood pH specifically, but any and all body fluid pH of less than 7.35 is acidosis. And this pH is generally going to be in the alkaline range. It's rare to see a pH less than 7.0, but it is more acidic than the normal range. So acidosis develops when there's more hydrogen than is necessary, or when there's more hydrogen that can be bound, or when there's not enough bicarbonate. And so this causes neurons to become less excitable, leading to signs and symptoms that are like nervous system depression, like slurred speech, drowsiness, unresponsiveness. And if it's left untreated, acidosis can lead to coma or even death. And there are different causes of acidosis, which are broadly divided into two categories: respiratory acidosis and metabolic acidosis. And without, any acidosis, the body's response is generally the same: decrease the amount of hydrogen and increase the number of base ions, especially bicarbonate. So if we look specifically at respiratory acidosis, we're looking at a decrease in the pH of body fluids due to excessive carbon dioxide. And this excessive carbon dioxide tends to happen when ventilation decreases, when a person is hypoventilating. And this can lead to an accumulation of carbon dioxide because less of it is being exhaled. This interferes with the carbonic acid bicarbonate ion buffer system, and it causes a shift toward excessive carbonic acid. And that excessive carbonic acid lowers the pH of the blood. And there are three general causes of respiratory acidosis: suppressed ventilation from brainstem dysfunction, blockage of the air passages in the lungs, or decreased gas exchange in the alveoli. Whatever the cause, the respiratory and urinary systems attempt to compensate. And respiratory compensation tends to begin within minutes of this pH disturbance, as hydrogen ions stimulate chemoreceptors in the brain. Okay, they say, oh my gosh, there's too much hydrogen, it's too acidic. These neurons then trigger an increase in the rate and depth of ventilation in an attempt to rid the body of excess CO2 and thus hydrogen ions. This is why an individual in respiratory distress initially causes hypoventilation will generally have an increased respiratory rate when they're actually in this respiratory distress, because it might seem like this individual is breathing rapidly to catch, because of an inability to catch their breath, but it's actually because there's an elevated amount of CO2, not low oxygen. And respiratory compensation for respiratory acidosis is not terribly effective because disordered ventilation was the initial cause. Such cases therefore rely more on renal compensation. And so the kidney tubule cells will absorb all of the available bicarbonate ions out of the filtrate and secrete extra hydrogen ions, producing new bicarbonate ions from glutamine. The renal compensation takes several hours to several days to have a significant effect, so these actions by the kidneys won't immediately be noticeable. So metabolic acidosis is really defined as the addition of hydrogen ions to the extracellular fluid from any acid other than carbonic acid, really. This term refers to the fact that it can result from acids, like metabolic acids, like lactic acid, uric acid, or ketones. And there are a number of different causes, like prolonged diarrhea leading to a loss of bicarbonate through feces, excess production of metabolic acids, like when a person is in diabetic ketoacidosis, failure of the kidneys to reabsorb bicarbonate, or failure of the kidneys to secrete hydrogen, and the ingestion of acidic drugs or toxins like methanol, wood alcohol. In any case, the first compensatory response is respiratory compensation, which occurs in the form of hyperventilation. In fact, hyperventilation in the absence of overt respiratory distress is one of the main signs that can point a doctor toward the diagnosis of metabolic acidosis. And the extent to which renal compensation really occurs depends on the health of the kidneys. If renal failure is the cause of acidosis, the kidneys aren't going to be able to contribute to compensation. However, if the cause is unrelated, the kidneys will work overtime to eliminate hydrogen ions and retain bicarbonate, as you guys saw with respiratory acidosis. Now, we've just established that the respiratory rate increases in metabolic acidosis as part of respiratory compensation. But how does changing the level of CO2 in the blood help when the problem is metabolic in nature? The answer is really that respiratory compensation doesn't correct the actual cause, but it corrects the resulting pH imbalance. Remember that the pH of a solution depends only on the number of hydrogen ions present, not the source of those hydrogen ions. So if you take a look at the following graphic here, you'll see that two hypothetical samples of blood, the one on the left is a normal pH, and the one on the right is a person suffering from metabolic acidosis. You see the purple hydrogen ions, and those are from carbonic acid, and then you see the white, or the blue rather, metabolic acids, and those are the hydrogens for metabolic acids. Do notice that the normal sample on the left has 20 hydrogen ions, 15 from carbonic and 5 from metabolic, with a pH of 7.4, right there in the middle of the normal range. The metabolic acidosis sample has 30 hydrogen ions as the result of excess metabolic acids and has a pH of 7, so it's very acidic for blood. Now, let's see what happens when this person starts hyperventilating and exhaling more CO2. So, as the number of CO2 molecules are exhaled increases, the number of hydrogen ions in the blood decreases, and you can see that here on the left. Or I'm sorry, you can see it on the right. Notice that the sample of blood now has 20 hydrogen ions, 15 from metabolic acids, 5 from carbonic acid. At this point, it's the same number as in the initial tube with a pH of 7.4. So we've compensated for metabolic acidosis. The condition that caused this acidosis has not been resolved, as shown by the fact that there are still a large number of metabolic hydrogen ions. But the number of hydrogen ions has gone down thanks to this respiratory compensation. These diagrams are super simplified and the pH values aren't really realistic, but the basic principle still stands. Respiratory compensation for metabolic acidosis works by lowering the total number of hydrogen ions in the blood. And the same situation exists for metabolic alkalosis, but in reverse. CO2 is retained to increase the total number of hydrogen ions in the body, returning the pH to normal. Alkalosis, not to be confused with alkalemia. Again, alkalosis is body fluid, alkalemia is just blood. But alkalosis is a body fluid pH of greater than 7.45. And this occurs when there's more base in the blood or more base in the body fluids than the buffers can handle, or when there's not enough hydrogen and the body fluid, the number of hydrogen ions in the body fluid lowers and decreases. Alkalosis basically enhances the excitability of neurons, so they fire action potentials inappropriately, and this results in sensory symptoms like numbness and tingling, motor symptoms like twitches or tetanic contractions, and if it's untreated, alkalosis can lead to seizures and death due to the sustained contractions of the respiratory muscles. And as with acidosis, there are two types of alkalosis. There's respiratory alkalosis and metabolic alkalosis. And the compensatory response for both involves the excretion of bicarbonate and the retention of hydrogen. So we're getting rid of bases and holding on to acids when we're in alkalosis. We're getting rid of hydrogens and holding on to bases when we're in acidosis. It makes sense. If your body fluids are too acidic, you want to hold on to bases. If their body fluids are too alkaline, you want to hold on to acids. Now, respiratory alkalosis is caused by a loss of CO2 through the lungs due to hyperventilation. Generally, this results from psychological states that increase the rate of breathing, but it can also occur when a person is at extremely high altitudes and dissolved oxygen in the atmosphere triggers this hyperventilation. If it's psychological in nature, there generally isn't much of a metabolic response. Usually the pH of the blood becomes too high and the individual loses consciousness, at which point the respiratory centers slow breathing and return pH to its normal range. However, if it is physiological in nature, the compensatory response involves the kidneys excreting bicarbonate ions and retaining hydrogen ions. So we're getting rid of bicarbonate bases, holding on to these hydrogen acids. These hydrogens that are produced by acids. And metabolic alkalosis tends to result from a loss of hydrogen ions or an excess of bicarbonate ions. And hydrogen ion loss is commonly the result of excessive vomiting, and so the excessive loss of the acidic stomach contents leads to this lowering of body fluid pH, or the raising, of body fluid pH. It can also be induced by the taking of certain diuretics or excess aldosterone, both of which lead to increased sodium reabsorption, and as there's more sodium being reabsorbed, more hydrogen is being secreted. And there is a possibility that excessive bicarbonate ions have been ingested. The culprit in this case is typically oral antacids taken for stomach ulcers or severe acid reflux. And the compensation for metabolic alkalosis is both respiratory and renal. Respiratory response is hypoventilation, retaining CO2 and giving carbonic acid a chance to be formed, increases the hydrogen ions in the body. The renal response is the same as it is for respiratory alkalosis. The kidneys hold on to hydrogen ions and get rid of bicarbonate ions in the urine. Now, acid-base imbalances can be difficult to determine which is which through standard blood tests, which normally, if you draw blood, you're drawing it from a vein. So instead, we look at arterial blood, and we determine this in arterial blood gases, ABGs. Blood is going to be drawn from the radial or the brachial artery, and the pH, the dissolved CO2, and the concentration of bicarbonate ions are all measured. From these values, it's possible for the clinician to determine the type of acid-base imbalance and the degree of compensation that has occurred. If no compensation has occurred, the imbalance is uncompensated. If some compensation has occurred, but the pH is still abnormal, this is a partially compensated imbalance. And if the pH of the blood is normal, but dissolved CO2 and bicarbonate ion levels are abnormal, it is fully compensated. And each acid-base disturbance has a characteristic set of ABG findings. Respiratory acidosis typically is shown with an elevated arterial dissolved CO2 and a lower blood pH. If the bicarbonate ion levels are elevated, then that means that renal compensation has begun and acidosis is partially compensated. Metabolic acidosis, the values for metabolic acidosis are slightly more complex, but the general rule of thumb is you'll have a lower pH and a lower bicarbonate ion level in the blood, but it's not always the case. Dissolved CO2 is also generally lower than normal because of respiratory compensation. And then due to the rapid effects of respiratory compensation, most cases of metabolic acidosis are partially compensated. And you've also got respiratory alkalosis and metabolic alkalosis. The main findings with respiratory alkalosis are pretty straightforward. High blood pH, low dissolved CO2. If bicarbonate ion levels are also low, it means that it's partially being compensated by the kidneys. And then metabolic alkalosis shows high pH and a high bicarbonate ion level. If respiratory compensation has occurred, CO2 levels will be elevated. And keep in mind that these are just sort of guidelines. Many cases of acid-base imbalances are complex and don't really fit these profiles. You can take a look here at the summary of the acid-base disorders, and it has their characteristic blood findings without compensation, their causes, and their main methods of compensation. I've already gone over these, but feel free to pause this presentation and review here. We're going to be moving on now. So up to this point, you've seen that the maintenance of fluid, electrolyte, and acid-base homeostasis requires the input of the two systems whose primary responsibilities are the maintenance of homeostasis. We need the endocrine system and the nervous system. And you've discovered that control of all three of these variables tends to be intertwined. All three have to be balanced together because imbalance in one can cause imbalance in others. If we look at a common imbalance, let's say dehydration from inadequate fluid intake, you see that it has cascading effects. We see first, total body water decreases, right, leading to imbalances of electrolytes and extracellular fluid pH. There's a decreased blood volume, possibly blood pressure, increased ECF electrolyte concentrations, especially sodium. So that leads to an increased osmolarity of the ECF, which draws water out of the cytosol of the cells, crenating them. And it also increases concentrations of metabolic acids in the ECF. There's less water molecules, and so the concentration of acid goes up. Metabolic acidosis may develop in this case. So after that, you've got juxtaglomerular cells releasing renin, which leads to the formation of angiotensin II, because the lower blood pressure and lower blood volume trigger that renin release, and so angiotensin II is the end product. And angiotensin II has several effects, the most immediate of which is to increase blood pressure through vasoconstriction. It also acts on the proximal tubule, increasing fluid retention of the kidneys, right? And by the main method it does that is it stimulates sodium ion reabsorption. Finally, it also stimulates thirst, which is going to lead to increased water intake if conditions permit. Several stimuli, especially angiotensin II and the increased osmolarity of the ECF, cause more antidiuretic hormone to be released, and this is going to dramatically increase water reabsorption from the distal tubule and the collecting system. And angiotensin II is finally going to stimulate aldosterone release. Aldosterone triggers that sodium ion reabsorption, and water follows it. It also stimulates potassium and hydrogen ion secretion, and so this hydrogen ion secretion helps to restore the normal pH of the blood. All the actions of angiotensin II, antidiuretic hormone, and aldosterone in combination increase the amount of water in the body. This restores total body water, blood volume, and blood pressure, thus decreasing the osmolarity of the ECF back to normal, and the increased amount of water also restores the normal sodium ion concentration. And although the mechanisms that cause sodium ion resorption, and more water molecules are reabsorbed than sodium ions. So this concentration is still normal. Finally, the excretion of hydrogen ions is going to restore the normal pH of body fluids. When these imbalanced variables finally return to their normal ranges, the secretion of angiotensin II, antidiuretic hormone, and aldosterone fall to their previous lower levels. And if you remember, this process shows how correction occurs under ideal conditions, and that some cases of dehydration are either too severe or too complicated for these mechanisms to resolve fully. But if you look at this next, so this slide shows that full response, the physiological response to dehydration, starting with, the lower blood volume, blood pressure, increased sodium concentration, increased osmolarity, and increased concentration of metabolic acids, all the way down to angiotensin II release, juxtaglomerular cells releasing renin, vasoconstriction, sodium reabsorption, thirst stimulus, antidiuretic hormone secretion, and aldosterone secretion, all of them working together to restore normal body water, blood volume, blood pressure, sodium concentration, osmolarity, and blood pH. And this discussion, we can determine from this discussion that the mechanisms for correcting water imbalance also correct the electrolyte and pH imbalance. So not only are the imbalances of each variable tied together, but the mechanisms that correct them are also linked together. So it's important to keep that in mind. That's all we have for chapter 25. Do not forget any of this material for the test, which should be coming up soon. If you have any question at all, you know my email, you know how to contact me. I will see you guys in the next one.